Welcome to an IXCO webinar, Neurological Disorders, Harmonization of Imaging in Clinical Trials. I have the distinct honor of introducing three of my amazing colleagues that I get to work with on a daily basis. But for you folks, you don't get to see them every day. And if you do get to see them every day, it's because you're working with us as a partner. So thank you so much. I'm just going to introduce our panelists. So our first panelist is Nicolas Fouin, Senior Biomarker Scientist at IXICO, who is an expert in neuroimaging acquisition and biomarker science, and he's currently a Senior Biomarker Scientist at IXICO. He holds a PhD in medical physics applied in nuclear medicine from UCL, and he has over a decade of academic experience and through his work as a researcher at UCL, MGH, Harvard Medical School, and King's College London. He's published numerous research papers on MRI and PET imaging acquisition, and he's been involved in developing guidelines for harmonizing MRI data acquisition and shared his expertise at international conferences, focusing on imaging protocol design and biomarker integration. I'm just going to check to see, because I think I can hear people in the background. So if you've got, you shouldn't be able to, just in case, please make sure if you, there should be no mute mics on, but if there is, obviously do apologize for that. If you can turn it off, that'd be fantastic. Then for the second person that I'd like to introduce, Helen Crawford, Head of Image Management at IXICO. Helen is an expert in neuroimaging and clinical trials and currently heads up our image management function at IXICO. She holds a PhD in clinical neuroscience from UCL and has over 15 years of experience in the field of neurology. She oversees the coordination and management of all of the activities required to qualify, receive, and analyze data for IXICO's clinical trials that we work in partnership with, and from startup to closeout. She has a clinical neuroscience background and a significant operational clinical trials experience. She has provided advice, consultancy, and input to sponsor trial protocols and practical critical tasks of supporting imaging sites in their scanner setup. So Helen's very much in the weeds of understanding what it means to harmonize imaging, and I can tell you that's a lot of information there. And then last but not least, our VP of Operations Services, Silke Grutong. She will be moderating our Q&A session, and she is approaching 10 years of tenure at IXICO, and she's currently the VP of our Operations Services. She's responsible for data management, radiology management, and imaging applications functions. She holds a PhD in medical imaging from the University of Surrey and an MBA at Imperial College, and she has over 30 years of experience in PET and MRI imaging across academic, industry, and healthcare settings in Europe and US. So you're in really capable hands here, folks. Lots of great experience here. We're going to walk you through imaging harmonization, and hopefully you're going to walk out here feeling very confident and being able to understand what it is you could potentially be doing and how you can apply imaging endpoints to your trials. Before we start, though, what I want to do is ask you a question. Which do you think are the biggest challenges in harmonizing an imaging protocol? So I'm just going to launch it now. If you can, or we should be able to see it now, is the poll box. So we are going to, hang on, let's see. We're launching the poll now, and just going to get some of the answers up. So the biggest challenges in imaging endpoints. So A, is it choosing the best imaging modalities? And that's really dependent on the therapy, your disease indication or therapeutic area. Is it selecting a suitable set of imaging sequences? So which imaging sequences are you looking at, and how which one's going to be working for which endpoint you're looking for? Harmonizing acquisition parameters. That one's a black box in its own, so we won't get into it in too much detail. But obviously, if you understand and know what that is, then you think, oh, is that a big challenge itself? Or is it finding the reliable endpoints from a non-harmonized data set? So that's in itself understanding what the ins and outs are between the data sets and what are the gaps missing, which doesn't fit. So I'm just going to see now where we are with the poll. So tools. 1 second, expanding. There we go. So I could see here that so far, out of our attendees, we're really saying here that finding reliable endpoints from non-harmonized data sets is the biggest challenge, which is great actually. Remember, this is multiple choice. You can if you want to, you can select all of them because there's no right or wrong answer to this. What is actually more important is actually being able to understand where you find the pain points all of this is. 63% of the attendees are finding that the reliable endpoints from non-harmonized datasets is a challenge, followed closely by at 51% harmonizing acquisition parameters. So actually, not attendees, it's just the votes. So the 75% of you have voted so far, and the way that the poll is split out is quite interesting. So I'm going to give it another 10 more seconds before we close out the poll. But I think to the panelists, this is going to be really helpful for yourselves and sort of like steering the direction of what you want to drive when you're talking about your when you're going through your presentations, because obviously like reliable endpoints is going to be the challenge here. I'm going to end and share the results with everybody on screen. So what we're seeing here, 64% is the endpoint, of course, followed by the harmonizing the acquisition parameters. That makes perfect sense if you think about it, because in the grand scheme of collecting all this information, longitudinally is probably the biggest challenge you're going to experience, collecting it over a period of time across lots of different geographies if you've got a huge multi-center trial. But if you've got a small trial as well, it's the same scenario too. No matter what it is, they've got their complexities. We're going to close that poll and we're going to go to the next one though. So we go to this next question, and I'm just going to open it now. What do you think is the biggest challenge for sites in harmonizing their multi-center neuroimaging studies? So this one here is a single answer question. Okay, so the first answer is setting up harmonized imaging sequences is the most challenging bit for sites. The second answer is consistency of image quality over time. Third, handling scanner changes or upgrades. And then the last one is real-time image quality control of scanner parameters. Now, each of them have their own challenges in their own way, and it is purely dependent on what your experience has been and also what you're looking to potentially achieve in the future, because each study in neurological disorders is so unique. They're very heterogeneous in that sense, but think about the ones that really apply to you, because that's going to be helpful in steering the conversation that Niccolo and Helen will have as they're going through their presentations. And we're going to stop there, and we're going to end and share the results now. So consistency of image quality over time. I'm actually not surprised by that, and panel. I don't know if that's surprised that you're probably nodding your head, thinking the same thing too. So yes, that's probably the most challenging for imaging sites, and of course, secondly, handling scanner changes or upgrades, because these things are the scanners are humongous beasts, so they will change depending on what it is, and it's not just hardware, it's also not just software, it's a combination of both. So yes, I'm glad that you've all answered these questions. I'm now going to close off the poll, and I'm going to go straight in to our first presenter's presentation. So Niccolo, I'm going to hand it over to you, and you're going to help us go through protocol harmonization in clinical trials. Take it away, Nico.
Thank you, Jenny. I'm Niccolo Fuin, and good afternoon, good morning to everyone. So as the next slide, please. So this talk is about protocol harmonization, as Jenny introduced. So as part of this talk, I will first provide a definition of imaging protocol harmonization and explain why it is important in a clinical trial setting. I will then discuss about how to choose the best modality for your study. This does depend on the sequences you use. That you use are for safety, eligibility, or quantitative analysis. It also does depend on which modalities are best for the specific therapeutic area and also on the specific phase of the study. The last section of this presentation will instead focus on practical approaches to harmonization. I will discuss how to standardize protocol parameters. I will introduce initiatives such as consortia like ADNI or PPMI, and I will provide some example of MRI and PET harmonization. Finally, I will briefly discuss post-adjustment methods that will be, however, the topic of a future webinar. Next slide, please. So the first section is, as I said, like an introduction on image harmonization and its importance in clinical trials. Next slide, please. So what do we mean by multi-site protocol harmonization? Protocol parameters harmonization refers to the process of standardized imaging acquisition protocols across different imaging centers to minimize the variability and to ensure that the data collected from multiple sites is comparable. This process is crucial to reduce biases introduced by differences in scanner hardware, software versions, and acquisition settings. The harmonization process entails the development of standardized MRI and PET protocols and port these to all needed platforms of the major scanner vendors. It also implies qualify all scanners at baseline and qualify following upgrades and perform real-time quality control. I will discuss the first two topics, whereas like the last two items will be discussed mostly by Helen in the next talk. Next one, please. So why is imaging protocol harmonization crucial for research and for clinical applications? So reduced standardization can have effects on the quantification and reproducibility of findings, and it had consequences in two case scenarios. The first scenario is the pooling of multi-site neuroimaging datasets, potentially acquired at also different times. The second scenario is how we relate new endpoints acquired under different scanning conditions to an existing set of normative data. The figure on the right shows results from the Oxford-Nottingham harmonization dataset, where 10 healthy volunteers were scanned on different 3T scanners. Each data point represents the median across all endpoints categories between a pair of sessions. So we can see, as on the right, the distribution of the within subject within scanner similarities is presented. In the middle, we have like the within subject between scanner similarities, which show variability that are similar to the between subject within scanner similarities on the left. This shows, so with this figure, I wanted to show that at the extreme, variability of measures obtained from the same subject, but on different scanners, can be as large as biological differences between subjects. So next slide, please. In this second section, we will discuss which modalities are best for your study, depending on the specific use of the modality. So next one, please. Which modalities are best for safety, eligibility, or quantitative analysis? A set of MRI images are normally selected for radiological read and safety purposes. For example, T2 FLAIR is used to identify wave matter hyperintensities, or T2 STAR is used to detect cerebral microbleeds. And these are safety images. We have a list where we have 3D weighted, T2 FLAIR, T2 weighted, T2 STAR, are all safety sequences. Additional sequences or modalities could also be taken in consideration for eligibility purpose. Here I give an example of the DAT scan that can be used in PD, so in Parkinson's, as it provides evidence of dopaminergic system integrity. And then there are exploratory sequences for quantitative analysis. These are dependent on the specific therapeutic area and on the mechanism of action of the drug, and are normally selected after review of the clinical protocol. So next one, please. The second consideration that we need to make is obviously choosing the modality depending on the specific therapeutic area. And this table here tries to summarize examples of modalities that are used in different CNS disorders. For example, in AD, in Alzheimer's, T1 structural MPage is used to study brain atrophy. More advanced MRI sequences, such as diffusion MRI, resting state fMRI, or perfusion imaging, are also normally used in AD clinical trials. Amyloid and tau PET are also very important modalities to include in AD trial. So in general, an AD trial normally makes use of both MRI and PET scanners with all the logistical and organizational complication of sort. Another example at the bottom could be the multiple sclerosis, where the T2 weighted structural FLAIR images are used to study MS lesion load, and additional sequences modalities which are not normally present, such as spinal cord or optic nerve images become also very important. Next one, please. So an important consideration to make when defining your imaging protocol is obviously the phase of the trial. Phase 1 trials have the objective of assessing safety, tolerability, and pharmacodynamics of the drug. They are small trials, often on few sites, and they need advanced modalities. And they also allow for better data harmonization. Phase 2 trials have instead the objective of determining optimal dose and gather preliminary efficacy data in a smaller cohort, in a relatively small cohort. They are medium-sized trials. They still allow for advanced and exploratory acquisition, but in these, a core set of modalities that can then be scaled up to phase 3 must be defined. Phase 3 trials are used to confirm efficacy and safety in a larger patient cohort and support regulatory approval. These are large trials with high variability of scanner model, manufacturers, and software versions. So here we need robust, very available on all sites, and well-validated sequences. And so because this needs to be deployed on a large number of scanners. Next one, please. The last consideration I wanted to make in terms of the sequences you select is the fact that obviously you need to take into account the duration of the protocol. Shorter acquisitions have the benefit of reduced patient discomfort and reduced patient motion. The table on the right shows a list of the sequences used in an exemplary PD trial with the corresponding acquisition time per sequence and also the total acquisition time of 46 minutes. Another consideration to make on top of the time is that when different modalities is required, such as in PD, for example, that spect is used, 2 acquisition sessions at different scanners are needed, with all the logistical and organizational complication of organizing the trial with 2 modalities. And next one, please. I will now introduce approaches to harmonization. So here I will speak about pre-scan strategies to harmonization. I will then provide some actual examples of harmonization in MRI and PET. And finally, I will briefly mention methods for post-scanning adjustment of endpoints, but however will be the topic of the future webinar. So next one, please. So strategies for multi-site harmonization. So these include the definition of a standardized set of scanning parameters and ensure that these parameters are as consistent as possible across all scanners. This is a non-trivial task that requires testing our real scanner with both phantoms and or healthy volunteers. Then there are vendor-specific adjustments are also needed because different scanning vendors may have different hardware and software. It's very important to work closely with vendors or, as in our case as well, a network of academic sites to identify equivalent parameters across scanner types. It is also important to perform phantom-scan at regular intervals at all sites. At least sometimes it's important to do so, and you can then compare metrics such as signal-to-noise ratio or geometric distortion to detect site-specific biases or drifts over time. Finally, it is important to perform quality assurance over time. This will be discussed more in detail by Helen during the next talk. And this includes, but is not limited to, monitoring scanner parameters over time and track scanner changes as well. Next one, please. So as I said, defining a standardized set of scanning parameters and ensuring that these parameters are successfully ported to all manufacturers' models and software versions is a huge task that involves a lot of testing and scanning of healthy volunteers' data on many scanning models and software versions. In these regards, an immensely useful role is played by the imaging consortia for the harmonization of imaging, that offer standardized guidelines and harmonized protocols. I here show like a list, a non-exhaustive list of consortia that we collaborate with the corresponding image modality. I don't have the time to go through all of them, but we have ADNI for Alzheimer's disease, PPMI for Parkinson, TRACK-HD for Huntington, TRACK-FA for Friedreich ataxia. Other one we collaborate with are DISCA for SCA. And other important initiatives are the Human Connectome Project, UK Biobank, very famous as well, OASIS or Magnis for MS. Next one, please. So MRI harmonization involves, we go to the example of MRI and what MRI harmonization involves. It involves that ensuring the acquisition parameters are as consistent as possible across all scanners. Core MRI imaging parameters include the repetition time, echo time, flip angle, voxel size, or bandwidth. For advanced imaging protocols, there are additional considerations that you might want to, that we need to make, so it is important to match the number of direction and diffusion gradients in diffusion MRI, for example, or for example in I don't know, perfusion imaging, in ASL perfusion imaging, it is important to align perfusion parameters such as labeling duration, labeling distance, or post-label delay. The figure on the right shows acquired multimodal data for a single subject across 6 scanners and 5 imaging modalities. From these images, you can visually see that, for example, the MPDage has been a well-harmonized sequence over time across the years and across the scanners, where there are still some differences in images from more advanced sequences, such as diffusion MRI. This figure is again from the Oxford Nottingham Consortium for Image Harmonization, which is a very nice paper from Waddington et al. In 2023 that I recommend. But anyway, to calibrate parameters across scanner, a common strategy is to adjust the parameters to accommodate the lowest common denominator. However, this is not a trivial task, but it's not always scalable as well, because vendor-specific proprietary implementation can often lead to only nominal matching of parameters rather than true matching, causing signal or contrast differences. That's why it's important to test them in a scanner with healthy volunteers or phantoms. Next slide, please. Here is a slide about the importance of protocol harmonization. So with the purpose of shedding some light on the importance of imaging protocol harmonization, traveling human phantoms dataset were collected for multi-site neuroimaging reliability studies. There are 3 recent very interesting projects which are cited here, and I'm showing where we use exactly traveling human phantoms on many scanners. And I showed just some examples, but I recommend to have a look at these papers because they're very nice. So in the first example on the right, on the left, sorry, 86 MPDage scan of the same healthy volunteer were acquired on 28 different 3 Tesla units over the course of 8 years. So the bar plots on the left are showing the median standard deviation of the total brain volume across vendors, according to changes in what we can think of the main parameters in MRI, which are resolution, echo time, and repetition time. So basically, these bar plots show how much the standard deviation of the whole brain volume, which is an important measure because it's the reference to many measures in the brain, changes dramatically when the main MRI parameters are changed by a factor of 5 or 10%. So here we wanted to highlight the importance of quality assurance to check that the parameters are not changed during the course of the study. On the right image, we have DTI scans of 205 healthy volunteers, which were acquired on 2 different 3 Tesla units. So the figure shows fractional isotropy and mean diffusivity values for white matter voxel for each healthy volunteer. The second column shows a scatter plot for fractional isotropy and mean diffusivity among the 2 sides. And the box plot also shows the differences in FA and MD in 2 co-regions, which are the left cuneus and the left putamen. And with this figure, I wanted to highlight that instead that DTI metrics are still not fully harmonized across all scanner models. Next one, please. So again, on challenges in the in MRI protocol harmonization, there are still challenges in the sense that, as I said, for example, in the first row, the images show an MPDA image of the same subject acquired with 6 different scanners, and as you can see, MPDA is a well-harmonized sequence. As I said in the previous slide, it's different for DTI in the sense that if you look at the second row visually, in this case, the DTI implementation, the same DTI implementation is available on all scanners. However, there are still some variation in image quality due to hardware differences. And even like an extreme case, I would say, is for arterial spill labeling, ASL, where manufacturers have been using fundamentally different sequences, and this raises obvious issues related to multi-site harmonization. So the first row shows a pulsed ASL CBF images acquired with the Siemens scanner, and the second row pseudo-continuous ASL CBF images acquired with Philips or GE scanner. And as you can see, they're completely different depending on the post-labeling delay time. Next slide, please. So again, reinforcing the concept, pulsed ASL and pseudo-continuous ASL methods differ fundamentally in both the spatial extent and the duration of the labeling. So pulse uses a single short pulse with a total duration of typically 10 to 20 milliseconds to invert a thick slab of arterial water spins in the neck. In PCASL, so pseudo-continuous ASL, labeling occurs over a longer period, typically 1 to 3 seconds, as blood flows through a single labeling plane and is inverted by an effective continuous RF pulse. So the two sequences have evident differences in image quality at the same post-labeling delay time. So you can see that, and as you can see from the figure on the bottom right. The figure on the top just wanted to be a little history of IOSL has been developed over the years with PASL and PCASL are two different basically branches of the development. So the fact that the different manufacturer so use different sequences types, so either PASL or PCASL, raises obvious issues related to multi-site harmonization. The solution we implemented in this case was to rely on WIP sequences, so research sequences, but with all the related issues of obtaining the relative licenses to install the sequences at sites. So there are additional complications if sequences are not already fully harmonized. I would say if are not already at least the same sequence among the manufacturers. Next slide, please. Here we'll talk about PET as well. PET harmonization. So in PET, the endpoint metrics are standard uptake value and its ratio, SUV and SUVR, and they are affected by many factors such as the construction parameters, system specifications, and in many different system specifications, and injecting those as well. And this introduces for MRI an additional source of variability, reducing the statistical power of these PET semi-quantitative endpoints. I won't go much in detail about these in this presentation because there are so many, but the table on the right shows a list of factors related to PET quantification and the corresponding explanation of how these affected the SUVs. Several studies aimed at reducing PET inter-system variability have provided harmonized protocol. There is a list here. They involve the optimization of parameters with respect to a target quantitative image quality indicator, such as target image resolution, recovery coefficient, or SNR. To calculate these metrics, phantom scan with known geometries and activity distribution are typically acquired and compared to digital reference objects. So phantom scans in PET play quite an important role. Next slide, please. So the most used approach to explicit PET harmonization is through the use of an Hoffman phantom. The method is based on the acquisition of an Hoffman brain phantom at every site and the calculation of a specific optimal Gaussian smoothing kernel to achieve a target image resolution. The Hoffman phantom is constructed to provide an anatomical accurate simulation of the brain regions, and is designed to simulate a 4:1 gray to white matter activity concentration ratio, as in FDG brain PET scans of healthy subjects. So after acquisition of the Hoffman phantom, data processing includes the steps for defining a site-specific digital reference object, for estimating effective image resolution at each scanner, and then estimating the full width al maximum of corresponding post-smoothing Gaussian kernel to achieve a similar resolution across different sites. So yeah, in short, basically, we use the Hoffman phantom to obtain the same smoothing kernel, different smoothing kernel, but will obtain the same resolution among scanners. The figure on the right, the left panel shows example of the original unharmonized phantom PET images acquired on 4 different PET systems. So we're harmonizing PET images, and on the right you see like that harmonizing the PET images using this method will produce a comparable visual image quality irrespective of the PET system model or make. And you see also the corresponding line profiles on the bottom of the figure. So if interested in the topic of Hoffman phantom harmonization, we recommend reading the paper from Shekari 2023, which is cited on the bottom of the figure. And so this so you can look at endpoints results after Hoffman phantom harmonization in an Alzheimer's clinical trial we also worked on. Next and final slide, please. So as a final, so next one, please. Sorry, thank you. As a final remark, I wanted to say that in this presentation we introduced methods to perform protocol harmonization. However, attempts to standardize acquisition alone will most likely lead to aligned protocols, but with inevitable differences across platforms. Because even using the same raw dataset acquired using the same protocols, the variability in processing option can lead to significantly different endpoints and results. So the final remark, in short, is that harmonization needs to be considered at all points of the study, from study design and data acquisition, which is very, very important, and this is what we presented today, but also data processing and endpoint extraction needs to be taken into account when thinking of image or data harmonization. So for that reason, post-acquisition harmonization approaches have been defined, and they aim to basically remove non-biological variability while still preserving variance in endpoints associated with biological factors. These methods, they're divided in statistical methods like combat or machine learning techniques that use, for example, GANs, and will be the topic of the next webinar. Thank you so much.
Thank you, Nico. That was a lot of insight, I have to say. And you know what? I think it eliminates a lot of the various different dimensions and challenges that you'd have just alone with the science of the acquisition. So thank you. I really appreciate that.
Thank you.
I'd like to bring on board Helen now to continue with the next part of our webinar. So Helen, over to you as we introduce you to the harmonization of operational considerations.
Thanks, Jamie. So I'm now going to talk about some of the operational considerations for a well-harmonized trial. So let's move to my first slide, please. So in addition to a well-harmonized imaging protocol, what other factors should be considered in your clinical trial? So over the next few slides, I will cover what we think are 2 of the most important factors, the first being site selection and startup, and the second, image quality. I will also talk about some of the other operational factors that you should consider, and I'll finish with 2 examples. So next slide, please, Jenny. So the most important question to ask at the beginning of an imaging trial is whether or not the sites will be able to set up the imaging protocol. And this is ascertained through the site feasibility survey, which is usually handled by the clinical CRO. Now this will confirm what scanner the site has access to, including the manufacturer and software versions, but it's really important to confirm the key requirements in advance in order to optimize this survey. For example, if only certain field strengths or coil types are to be accepted, the specific parameters that Nicola was talking to us about, for different sequences, these can be specifically asked in the survey. And for any novel or more advanced sequences, you should ask the site if they have experience in acquiring those sequences so they can confirm whether they can support them or not. And during this feasibility stage, the imaging CRO may be able to provide a technical summary, which will outline all of the key parameters, which will help with site selection, as well as expedite contracting of the imaging centres. Now, a good imaging vendor can make this available once the full requirements are received from the sponsor and before the imaging manual is finalised. At this feasibility stage, it's also important to ascertain if there are any known scanner events that are to be expected, and these scanner events can vary in their impact and can be considered as major or minor. And what do we mean by that? So by scanner event, we mean, for example, a change in scanner hardware, change in the head coil, or replacement of gradient coils, and these can be major events. And software version changes are minor changes, but still need to be monitored, and manufacturers frequently perform software upgrades. So ideally, the imaging CRO should be aware of these events in advance, as any changes to a qualified scanner can affect data collection and image quality. So where possible, try and limit these events, but of course they're unavoidable. So therefore, if your imaging vendor is aware at the start of the study, they may advise selection of a different centre or see if the site has access to a different scanner. If necessary, the initial site qualification might be postponed after any major scanner changes have happened, still keeping in mind your first patient in timelines. And we know that one of the biggest challenges when setting up neuroimaging studies is ensuring sites are qualified quickly. But by optimizing the site feasibility survey and planning for scanner events, this will help select the right sites and will help speed up the site activation times. Next slide, please. So, as well as the site survey, there are other important activities during the start of a project. It was about 2 years ago now, our colleague Nicoletta Vitale, head of our project startup at Ixico, did an excellent webinar on how to succeed in neuroimaging clinical trials, and she explained in detail the site qualification process. This webinar is still available on our website, so I do recommend it. However, it's worth just briefly reiterating some of the points again now. So the imaging CRO will send a technical site survey to confirm details about the scanner, confirm the sites can support the protocol. It helps to make this survey easily accessible for the sites and available online. Similarly, the training for the sites should be available online, ideally self-paced, easy to follow, and importantly tailored to the user role. So for example, for those acquiring the sequences and then for those responsible for uploading the data to the data management system. Making the operations guide, the imaging manual, all materials available online will also help, as well as any other summaries or checklists that get produced, because this can really help the sites and make it easier for them to acquire the data, and send this to them as soon as you can. Identifying the right site contacts also helps prevent delays, as well as ensuring there's a trained backup at the site. So there should always be at least 2 people trained in how to acquire the scans and how to upload the data. And finally, having an online central platform for receiving and storing the data is ideal. This means the imaging CRO will be able to review the data, give timely feedback regarding quality, and also allows for remote site management and site support. And this centralized site management means you have a consistent and standardized process throughout the trial. Next slide, please. So the second most important factor after site selection and site startup is image quality. We know that another challenging aspect when setting up and managing neuroimaging studies is obtaining high quality data. And I think that came out as one of the top poll answers earlier as well. So it's really important to reiterate sites must acquire the scans in accordance with the imaging protocol with the same parameters for every visit throughout the course of the trial. Your imaging CRO will perform image quality control to confirm the scans are acquired in accordance with the protocol. This will involve a visual quality check to identify gross artifacts such as motion or poor patient positioning, and then will assess whether the data is suitable for radiological read and/or analysis. And a good imaging vendor will also perform a metadata check to confirm the parameters are correct and compliant. What also is really important is good communication between the site and the CRO to ensure the site is notified straight away if the data is not compliant or it's of poor quality, and whether or not a rescan or resubmit of the data is required. If image quality is poor, your imaging vendor should be able to offer retraining to the site or provide access to expert site managers and imaging scientists who can jump on a call to assist. A good imaging vendor will not only query the site for rescans and resubmits, but will also provide feedback to help the sites improve their scans and maintain good image quality throughout the whole study. So at ICSICO, we call this a scope for improvement query, and it helps ensure longitudinal consistency. Your imaging vendor should also train the sites ideally on how to perform a real-time quality check of the data whilst the patient is in the scanner, checking for motion or any artifacts, meaning that a rescan can be acquired in the same scanning session if necessary. And most imaging protocols will include 2 T1 weighted sequences, which increases the chance of a good quality structural scan being acquired in the session. These actions together will help reduce the likelihood of rescans, preventing the need to reschedule patients, reducing the burden to the patient and to the site, preventing delays in dosing, reducing scanning costs, and in turn will speed up safety and eligibility reporting times. So one final point is just about the importance of the baseline scan. When measuring change over time in a trial, this baseline scan will be used as the reference for all the follow-up visit data to be compared to, therefore is key for the longitudinal analysis. So good image quality and longitudinal consistency is essential for safety and efficacy. Next slide, please. Now, there are several other operational factors to consider. As well as confirming the sites can support the imaging protocols, the sites must have the suitable resources and staff availability to take part in the study. There must be adequate availability time on the scanner to book in the patients, but there also needs to be enough staff to provide good communication between the sponsor and vendors so they can respond to feedback in real time. We would advise asking your imaging CRO if they have any recommendations, if they've worked with any of the sites before and have any specific site recommendations. Now, personnel changes are going to be unavoidable, and will happen throughout the duration of a trial, particularly a long trial. So it's important to inform the imaging vendor in advance so that a replacement can be trained and their contact details shared in advance to minimize any risk that communications are lost. As I mentioned before, it's important to have at least 2 or more staff trained and available in both how to scan and upload the data. Also, as I mentioned previously, it's best to plan for any scanner events where possible, including scanner requalifications throughout the course of a study. Over a longer study, it's very likely that there'll be a scanner event, for example, a planned upgrade, a replacement, maintenance, etc., or a protocol update. If the imaging CRO knows in advance, then test scans, either a phantom or a healthy volunteer scan, can be arranged before and after the change, and then the CRO can then review this data. As part of the quality control, the imaging vendor will be able to detect major scanner changes, for example, if a different model or make of scanner was used. However, some changes in hardware, for example, change in the head coil or replacement of the magnet gradient coils, aren't detectable through the metadata checks alone. Software version changes are often minor and can be detected through the data checks, but these still need to be monitored. Now, we know not all events can be planned for, but having a good process in place and following the steps we've outlined will help with a quick requalification and prevent delays or disruption to patient scheduling. Ideally, the same scanner is used by each site for all subjects and for all time points, but we know this isn't always possible. So the most important thing is that any changes are tracked, and a good imaging vendor will record every time there is a change and will report this information to the sponsor when transferring the safety and efficacy data so that it can be taken into consideration when performing the analysis. And finally, it's important to reduce reader variance. Having taken the time and effort in harmonizing between scanners to minimize variance in the data, it's essential to make sure the data is then read in a harmonized way. This is a big topic and it's beyond the scope of this webinar to go into detail, so it may well be the focus for a future webinar, so watch this space. However, it's important to reiterate that you should have all of your data read centrally by expert radiologists and analysts who have been trained and validated to ensure good inter-rater reliability. Okay, thank you. So now on to my last slide. I'm just going to present 2 examples to demonstrate some of these key operational factors that I've presented. So the first example is a large-scale study a phase 3 trial requiring large-scale harmonisation. So this might involve standard safety and volumetric sequences. However, there will be multiple different scanners with different makes and models in many different geographical locations. You need to consider that there'll be many different site staff, so there'll need to be good coordination from your imaging vendor for all of the training and the qualification. And it's important to consider the numerous languages that will be needed, and a good imaging vendor will offer translation of the materials into these different languages. So the use of standard structural imaging with well-harmonized sequences, like some of those that Nicolo talked about, should allow the use of on-site available phantoms and quicker site activation. But also don't forget, these larger and longer trials will definitely have personnel changes and will definitely have scanner changes. And trials with large time intervals will also require periodic requalifications so that the imaging CRO can assess if any upgrades have occurred during these intervals. Now for the second example, imagine a smaller but more complex trial, perhaps a phase 1 with just 2 to 3 sites, but with more than one modality and with several advanced sequences. It's really important to specify the key parameters in advance and ask if the sites have experience with these sequences. You'll need to bear in mind that there'll be more than one scanner to qualify per site, and the use of bespoke phantoms may be required, and these may need to be sent to each of the sites. This will mean that site activation may take longer. Close collaboration with the CRA to facilitate good communication with the site will help. It's also important to note that with these trials, data will be acquired in multiple acquisition sessions, like Nicolo also mentioned. So depending on the number of different modalities, for example, if both MRI and PET data is being acquired, this will be done separately on 2 different scanners. So this will need to be considered when arranging patient visits, which will need to be carefully planned in the right order by the sites. So as you can see, there are many challenges to multi-site imaging, but hopefully Nicolo and I have reassured you that it's completely achievable with the right preparation and considerations. So thank you. That concludes my presentation. Thanks for listening.
Thanks, Helen. That was great. And I think it just really opens up a lot of human logistics and when you're putting in imaging harmonization and what's involved when you're putting the right place and the right hardware, software, and the positioning together. So without further ado, what I want to do now is I'd like to bring Silka and Nicolo back, and I want to go straight into questions because I'm pretty sure there's a couple of questions burning. And I can see the questions box, which you can submit your questions in at any time. It's quite full at the moment, but what I want to do though is hand over to Silka, who will take us away with questions.
Thank you, Jane. Helen, I'd like to come to you first because there's a question for you. You already mentioned dealing with the events of a scanner change or a scanner update, but there was a question here about managing changes in the study protocol and such as additional scans and imaging sequences being added. So how would that be handled mid-study?
So protocol changes do happen. And as I said before, with longer studies, it's something that you can't avoid. But ideally, the sponsor will give plenty of notice so that the imaging CRO can contact the sites to confirm that they can acquire the new sequences and allow enough time for the imaging manual and the site training materials to be updated and for sites to be retrained. Once the protocol is confirmed, depending, I suppose, on the complexity of the protocol, sites would need to send the imaging CRO an updated protocol printout from the scanner. If the protocol updates just involved removing sequences, then the printout would be enough. But if new sequences are added or advanced sequences are introduced, there would need to be another test scan or a phantom scan for the imaging CRO to review. And I guess with any of these protocol updates, the key thing is sites don't scan patients with the new protocol until they've been requalified.
Mm-hmm. Oh, thank you. Well, I will come back to you because there's another question already in the chat for you. But meanwhile, Nicolo, coming back to your presentation, I know you touched on this, but to conclude, in your opinion, are there any sequences that can't be harmonized? And furthermore, are there any makes of scanners that can't be harmonized or should be avoided? What approach is advised when dealing with less common makes of scanners?
So yeah, Thank you for the question. I provided an example of ASL, for example, during the presentation, where it can be fully harmonized the sequence using the product sequence, but you always have the chance of using work in progress sequences
Work in progress.
with all the relative issues about getting the right licenses to install them for the clinical trials. Other examples could be, for example, that I can think of so magnetization transfer prepare sequences where like the MT prepulse is difficult to, each manufacturer has a different shape of the prepulse, really, or power of the prepulse, and so it's difficult, they are difficult to be harmonized. And there are also sequences which are very much okay, when there is like a plethora of sequences which are still in the research setting, and they cannot be harmonized among manufacturers because there is no product sequence for any of the manufacturers. And I can think of, for example, very new sequences such as inhomogeneous, talking about MTI, also inhomogeneous transfer imaging, or some of those. The second question about instead
Scanner
the
models.
scanners. So obviously, like there are many scanner manufacturers that I didn't even know about. There are like more than 20. However, like obviously most of the effort in harmonization has been done, especially in the consortia which I mentioned, so like ATLIN, PPMI, has been done in trying to harmonize the three main manufacturers, which are GE, Philips, and Siemens. However, there are like some which are also growing in importance. Some manufacturers such as Canon and United, and they provide sequences which can be harmonized. It's just about having the contacts, as we do, to work with them. And I find also that like these sort of new ones, they also understand the importance of basically being in the clinical trial field, if you want. So they help most of the time. So yeah, it is the answer is like to a certain extent you can harmonize to at least 5 manufacturers, I would say, and then the other ones start to be a bit more complicated, obviously, because they're very niche.
So something
Scanners.
to consider doing site selection.
Site selection is definitely. I mean, the problem with the other ones, which are not the 3 main ones, is that there is not much normative data to compare to.
Not much, yeah.
So even even if you harmonize with even if you acquire with an harmonized protocol, still like all the normative data that you have from the past will be from the 3 main one. That's definitely a limiting factor, I would say.
Thank you. I'll come back. We have a few minutes. I'd like to come back to Helen because there's an interesting question here about a specific therapeutic area I think you would probably like to address. It's regarding our experience with movement disorders trials. What's the approach to maintaining image quality in that particular patient population, and in particular the potentially progressive challenges in maintaining the harmonized imaging in the longitudinal studies and in the imaging sequences?
Yeah, there's lots of different things we can do. Obviously at EXCO we have quite a lot of experience with this. And I think the first thing is to make sure your most important sequences are at the start of the scanning session. So your safety structural sequences are at the beginning of the protocol. So when the patient first gets into the scanner, those are acquired first. Like Nicolo touched on as well, it's important to consider the length of the protocol, the number of sequences that are required to try and sort of minimize that patient burden. I think if scans do have to be long in length, consider adding in an optional break so the patient has a chance to move around or get out of the scanner. But obviously with movement disorders, trying to limit the movement, so additional padding around the head, trying to make sure the patient's as comfortable as possible is important. I think as well what I touched on before about sort of the technicians themselves doing a real-time quality check in the scanner to see if they can acquire a second or even a third structural scan whilst the patient's still in the scanner. We often see that, and it really helps increase the likelihood of a usable scan. I think it's important the team reviewing the data are well trained and know what's acceptable for read and for analysis, focusing on the key regions of interest or the key biomarkers for the study, obviously usually more subcortical structures in movement disorders. But of course, if the patient is not able to rescan and we get the best that we can from a patient, then we would consult with our radiologists to make sure to see if they could actually read the scan before failing it, and we'd look to analyze as many of the endpoints as possible before failing the data. And it may be that some of the endpoints fail, but we do try and get as much from the data as possible. And then I think in some scenarios sedation is also allowed, which may help the patient keep still.
Yeah, and that will be specified in the protocol. So thank you, Helen, because that's a lot of practical tactics and approaches to optimize that. So Jane A has called time on our discussion. There are some other questions in the chat that I can see. We'll make sure that we respond to those offline if you've left your details. But back to Jane A now to close off our session.
Thanks, Silka. I feel really bad because it was actually really good. I was waiting. There's lots of great questions that have come through. So I want to thank the audience for actually submitting your questions through. And I really hope that it's been helpful for you in terms of being able to understand what's needed, and also some of the peaks and troughs of the complexities, whether it's the sequences, whether it's advanced or standard, and also the kind of operational lifts that you'll need to do when you're conducting phase 1 or phase 3 trials. So thank you so much, team. I appreciate that. As always, we're going to conclude this webinar by just saying that if you hang around for a bit afterwards, you'll be sent over a brief survey just to help, so it can help us guide future content and webinars. This webinar will be part of a series of continuous imaging harmonization operations webinars, and we want to know which direction you want to go in next. So there's been a couple of points that have been dropped down in Helen and Nico's presentation. So please make sure that you have your say by filling in the survey at the very end. You can also make sure to follow us on LinkedIn, as we're consistently dropping the latest news, including all of the latest advancements and the work that we do across neurological disorders research. And also, if you happen to know anybody that actually that benefits from this webinar and all the other webinars we've done in the past, please forward it to them. Otherwise, we hope you've enjoyed this talk and today's talk. We look forward to seeing you in another webinar coming up. Otherwise, from us here at Ixico, have a fantastic day, afternoon, evening, no matter where you are and what you're doing. Take care, everybody.
Thanks. Bye.